Okay guys, in this particular lesson we are going to focus on specifics of cellular division. So the first thing that we're going to cover is exactly what the cell cycle is, and the fact that the cell cycle is all of the changes that a cell is going to go through from the time that it forms, so meaning that the time that it is developed, obviously or typically, from another cell dividing, from that point in which it forms until it itself is dividing again to form two new cells. So the stages of the cell cycle are broken down into interphase, mitosis, and cytokinesis. And for those of you that remember this, mitosis should ring a bell, and you should be thinking PMAT. I, P, MAT, C, being the entire cell cycle. And this picture over here is just kind of showing you how a rather large portion of the cell cycle is considered interphase, and then we break down mitosis into that PMAT, and then we have that cytokinesis piece. And I'll go more into this picture later. So the first step for cellular division is interphase, and this is really going to be a period of growth and function. The cell is pretty much carrying out its normal functions and doing its normal things while it's growing. Interphase is broken down into the first growth phase, synthesis phase, and second growth phase. Also, sometimes you may see growth used interchangeably with gap. So you may see that it's the first gap phase, synthesis phase, and second gap phase, meaning the exact same thing. But typically, I really like to use growth because that focuses on exactly what's going on during this point in interphase. So if we look at the G1 phase, all right, first growth phase, basically, like I said, cell is maintaining its normal functions, carrying out its typical things, but it's growing. And that growth is really about creating one larger cell because it's preparing to divide into two, and obviously it needs to get bigger so that each of its daughter cells will be substantial in size. Then we have that S phase, the synthesis phase, and we should all know that synthesis means to make. So in this phase, something is being made, and what's being made is a copy of the DNA or the chromatin. It's going to be replicated. And then we have that second growth phase, and this is when the cell is going to copy all the other organelles. Everything else is going to be copied, the cell is going to grow a little more, and it's really going to be preparing for the nucleus to divide. So again, showing this picture, we've now passed from the start through the first growth phase where everything is growing and preparing, synthesis when the DNA is replicated, which you can see takes a pretty substantial amount of time, and then the second growth phase when other organelles are replicated and the cell continues to grow. Right? Now we're moving into the mitosis stage. Mitosis is something that's going to occur in all the somatic cells, right? Somatic, we're talking about body cells, right? Remember that there's a whole nother process called meiosis for gametes, for sex cells. Karyokinesis is really nuclear division, right? And that's mitosis. Mitosis is that nuclear division. Again, we're going through PMAT, prophase, metaphase, anaphase, telophase, in order to have the nucleus divide. This is just a picture showing the different stages of what goes through mitosis, and if you want to come back to this at a later point in time, please let me know, and I will be happy to provide you with a picture of this that's nice and clear. So, in prophase, some of the key characteristics are that chromatin is going to condense into the chromosomes, so we're going to go from being a typical coiled up strand to now we're really going to be overly coiled up, and that's going to create a thickness to make those chromosomes more visible. Nuclear envelope is going to disappear, so as you can see it's kind of starting to break down here. Centrioles are moving to opposite sides, right? So we have the original centrosome, right, and the copy of the centrosomes, the two of them, and they're actually going to opposite sides of the cell. And then we have spindles that are forming, and these are those microtubules that were a part of the cytoplasm's cytoskeleton that we talked about in the previous sections, right? So these microtubules are going to be what the spindles are forming out of. We can then go into metaphase, and what's going to happen is now that the spindles are spanning the entire length of the cell, they're actually going to reach out and they're going to grab onto these centromeres, the center portion of the chromosome, and they're going to pull the chromosomes in line in the center of the cell. Meta, middle, right? So that's when we're lining up in the middle. And then we have anaphase, and I typically associate ana, apart, right? In anaphase, what happens is these spindle fibers are going to kind of start to pull on the chromosomes in opposite directions. And that pulling force is going to bring them to tear apart, right? So you can see that what we had previously, right, together, has now been pulled apart. So the chromosomes separate and they're being pulled to opposite sides, and you can see how they're kind of starting to separate to separate sides, and what parts of the cell are going to become their own cells individually. Then we have telophase, and telophase is kind of like an opposite of what's going on in prophase, because in telophase the nuclear envelope is reforming and the chromosome is unwinding back into chromatin, so it's kind of undoing what was done in prophase, kind of backwards. So now we've got two nuclei that are forming, right? The spindles should be kind of starting to break down a little bit. We got our chromosomes are unwinding. Soon this will become invisible. And as you can see, we're kind of almost starting this whole, pinching in business that's going to take place. So now we've gone through prophase, metaphase, anaphase, and telophase, as well as all of interphase, and we're really reaching up to our point of cytokinesis. In cytokinesis, we're talking about cytoplasmic division. This actually really occurs during anaphase, and as you saw in the previous pictures during telophase, you could see that there seemed to already be some pinching in, that there seemed to already be a division that was beginning in the center of the cell. So it was already occurring in telophase, and that's because it begins in anaphase, as the cell membrane is pinching a little bit. Now you're, basically creating two cells that are going to each have their own set of organelles, their own set of inclusions, their own set of genetic material, so that they can become their own complete daughter cell. And that contractile ring, which you saw in the previous telophase picture, was really kind of like a line here, right? And it would have looped around to the other side, right? And what you would have gotten there would be a ring that was going to kind of contract and constrict, and as it contracted and constricted, it would pinch the cytoplasm in half, and that cell membrane would be pinched as well. So when all is said and done, the cell division results in two genetically identical cells, 46 chromosomes each. We should know that 46 gets doubled into 92, and when it splits back into 46 and 46 again. And this is just a perfect example under a microscope of cellular division taking place.